Assalamu alaikum. Good morning, everyone. Today, inshallah, we are going to talk about the exocrine pancreas. As you know, the pancreas is a retroperitoneal structure present behind the stomach, and it has two functions. It is considered as two glands in one. So it has an exocrine part and it has an endocrine part. And the endocrine part is mainly involved in the production and release of insulin that is very important for the glycemic control. The objectives of today's lecture are to recognize that the acinar cells produce the pancreatic enzymes and that the duct cells produce sodium bicarbonate and water. And this sodium bicarbonate is very important for the neutralization of the acidic chyme. Describe the physical properties of pancreatic juice, and then enumerate the main organic constituents of the pancreatic juice and describe their functions. Recognize that the main inorganic constituent is sodium bicarb, and then describe the neural and hormonal regulation of the exocrine pancreatic secretion. And finally, describe the three phases of pancreatic secretion: the cephalic phase, gastric phase, and the intestinal phase, something similar to the phases of gastric secretion that we already studied in one of the lectures. As you can see here in this illustration, this is the pancreas. It is composed of pancreatic acini that empty their secretions through a ductular system and that finally empty into the main pancreatic duct that opens into the duodenum at the ampulla of water. And the acinar cells are very active cells in terms of protein production. That's why they contain a lot of zymogen granules that release their content by exocytosis into the lumen of the pancreatic ducts. Also, between the exocrine acinar cells or pancreatic acini, we have pancreatic islets or islets of Langerhans that are composed mainly of four types of cells: the alpha cells, the beta cells, delta cells, and the F cells. And we'll talk about them in brief in the coming slide, inshallah. So the pancreas contains two types of glands: exocrine gland that represent 98 to 99% of the total pancreatic mass. The exocrine gland is called the pancreatic acini, which secretes digestive enzymes and bicarbonate into the intestinal lumen. The endocrine gland, or the endocrine portion of the pancreas, it represents only 1 to 2% of the total pancreatic mass. It is called the islets of Langerhans or the pancreatic islets. The normal human pancreas contains between 500,000 to several millions of islets or pancreatic islets. Islets can be oval or spherical, and they measure between 50 to 300 micrometer in diameter. Islets contain at least four types of secretory cells. These are the alpha cells that represent 17% of the total mass of the pancreatic islets, and these alpha cells produce the hormone glucagon. Beta cells, these are the main cells of the endocrine pancreas. They represent 70% of the total endocrine cell mass, and these beta cells produce insulin. Delta or D cells, they represent 7% and they release the hormone somatostatin. And finally, the F cells, they represent 6% of the endocrine pancreas and they release the pancreatic polypeptide. On the other hand, the exocrine pancreas is a compound alveolar gland resembling the salivary glands, which is a specialized secretory organ that contains a branching ductular system through which they release their secretory products. In case of salivary glands, saliva is the secretory product, while in case of the pancreas or exocrine pancreas, the product is the pancreatic juice. The principal functions of the exocrine pancreas are to neutralize the acidic gastric chyme that is delivered from the stomach to the duodenum during gastric emptying, and to complete the intraluminal digestion of the ingested carbohydrates, proteins, fats, and even nucleic acids. The pancreatic juice is composed of different components that are a product of two different cells. So the two different cells produce the exocrine pancreatic secretion. Number one are the acinar cells. They are grouped into lobules and they contain, or the granules containing the digestive enzymes or the zymogen granules are formed within the cells and then released or discharged from the acinar cells by exocytosis from the apical border of the cell into the lumen of the pancreatic acini and then into the small pancreatic ducts. The other part of secretion is released from the ductular and ductal epithelial cells. They secrete an alkaline fluid that is very rich in bicarbonate. The pancreatic juice flows along the main pancreatic duct, which empties into the duodenum along with the common bile duct that we already covered in the previous lecture. The pancreas or the exocrine pancreas produces about 1,200 to 1,500 ml of pancreatic juice per day. The pancreatic juice is isotonic compared to the blood. It has an alkaline pH of around 8 and due to the high bicarbonate content. The bicarbonate concentration in the pancreatic juice is between 140 to 150 milliequivalent per liter, which is relatively high compared to the concentration of bicarbonate in the plasma, which is about 24 milliequivalent per liters in the plasma, as I said. The pancreatic juice is a clear, colorless liquid consisting or composed of water. As we know, most of the secretions or body secretions are composed mainly of water. Number two, sodium bicarbonate, which acts as a buffer for the acidic chyme in the duodenum. Then we have many enzymes in the pancreatic juice. These are pancreatic amylase, proteolytic enzymes, trypsin, that is secreted as inactive enzyme, trypsinogen, and it is activated by enterokinase that is present in the duodenum, chymotrypsin, and it is released as chymotrypsinogen, the inactive form of trypsin. And we have another or third peptidase, which is carbopeptidase, released as pro-carbopeptidase, which is again inactive, and elastase that is released as pro-elastase. All of these are, as I said, secreted as inactive enzymes. They will be activated changed or transformed to the active form in the duodenum, or once they are released into the duodenum. Pancreatic lipase, the other enzymes are pancreatic lipase, cholesterol esterase, and phospholipase, and ribonuclease and deoxyribonuclease for the digestion of the RNA and DNA. The peptidases are released as inactive enzymes because if they are released as active enzymes, they will cause autodigestion of the pancreatic parenchyma. So this is just is meant to protect the pancreas from autodigestion after their release from the acinar cells. Now let's talk about the mechanism that is involved in the enzyme secretion. This is, or as you can see here in this illustration, we have part of the pancreatic acinus where we have one acinar cell, and this is the basolateral membrane and its apical membrane. The acinar cell contains zymogen granules that need calcium for their exocytosis or the exocytosis of their content. And also the apical membrane has chloride channels that are activated by an increase in the intracellular calcium. Okay, so neurohormonal factors like acetylcholine or CCK, they will stimulate muscarinic receptors or CCK receptors respectively. Both of them will lead to an increase in the intracellular calcium. The intracellular calcium will induce the exocytosis of these zymogen granules, and their content or their enzymatic or the enzymes inside those granules will be released into the acinar lumen. Moreover, the high calcium, as I said, can stimulate the apical chloride channels. But as we know that chloride inside the cell is or chloride concentration is smaller compared to the extracellular fluid. So what is the source of this chloride? The source of this chloride is a co-transporter, which is sodium potassium 2 chloride co-transporter. It is a secondary active transport system that depends on the primary active transport system, sodium potassium pump. This pump will generate the sodium gradient that will drive the secondary pump. So this pump now will pump in chloride. It will build up chloride concentration or chloride inside the cell at high concentration, and this will allow chloride to move into the lumen of the acinus when the chloride channels are opened by high intracellular calcium. The potassium will leave the basolateral membrane into the interstitium and will be recycled again through the co-transporter. The movement of chloride into the lumen of the acinus will create an electrical gradient that will bring sodium through the paracellular pathway to enter into the acinar lumen. Both of them are osmotically active, sodium and chloride. Both of them, they will drag water either transcellularly through the aquaporine channels or through the paracellular pathway between the acinar cells. So let's summarize in this slide. A rise in the intracellular calcium concentration produced by neurohormonal agents like acetylcholine, gastrin, and cholecystokinin will lead to the exocytosis of zymogen granules. The contents of the zymogen granules, and these are enzymes, are released into the acinar lumen as we have seen in the previous slide. The acinar cells also secrete small quantity of isotonic fluid to wash the enzymes into the duct of the tree of the pancreatic ducts, and this is achieved by the following mechanism. Number one, the increased intracellular calcium concentration produced by stimulants of enzyme secretion, and these are acetylcholine, cholecystokinin, and gastrin, will activate a protein kinase, and this will lead to the opening of chloride channels in the apical membrane. As we have seen in the previous slide, these will lead to chloride diffusion from the cytoplasm into the acinar lumen. The gradient for chloride diffusion is maintained by the influx of chloride from the interstitial fluid across the basolateral membrane by means of the sodium potassium 2 chloride co-transporter. As I said, this is a secondary active transport system. This transfer or this secondary transport system is driven by the sodium gradient that is created by the primary transport system or primary active transport, which is the sodium potassium pump. The electronegative lumen will attract sodium to move from the interstitial fluid through cation channels in the tight junctions between adjacent acinar cells. So sodium will pass through via the paracellular pathway. Water will move from the cells and the interstitial spaces to the lumen of the duct system to produce an isotonic secretion. And I said water can move through the paracellular pathway between the cells or through the cells using special water channels called aquaporins. We have seen that the acinar cells produce enzymes, all the digestive pancreatic enzymes, and a small amount of fluid that is rich in sodium and chloride. So what is the source of bicarbonate in the pancreatic secretion? The major site for the production of bicarbonate-rich juice is the duct system. So the pancreatic duct system. The composition of secretion varies with the flow rate. So at low flow rate, here we can see the secretory rate on the x-axis and the concentration of the 4 ions on the y-axis. This is the concentration of potassium, the red line. The light green line is the concentration of chloride, then the bicarbonate, the orange line, and the dark pink line is the concentration of sodium. We can notice that at all or at different secretion rates, whether low or high, the potassium and sodium concentrations won't change. Potassium concentration is very low in the pancreatic juice, while the sodium secretion is very high or equal to the plasma concentration of sodium. What changes depending or according to the flow rate is the concentration of chloride and bicarbonate. As the flow rate increases, or the pancreatic juice flow rate increases, the chloride concentration will drop, while the bicarbonate concentration will increase until it will reach a plateau. Okay, so as I said, the composition of the secretion varies with the flow rate. At low flow rates, the major ionic constituents are sodium and chloride. So at low flow rate, we have chloride high and sodium is high. At high flow rates, the predominant ions are sodium and bicarbonate. So this is the concentration, or the two ions that are predominant at high flow rate, which are sodium and bicarbonate. The concentration of potassium and sodium at all flow rates are similar to those of the plasma, as we can see here. They are stable and they are equal to their respective concentrations in the plasma. The concentration of chloride falls and that of bicarbonate will rise as flow rate increases. Here we can see an illustration showing a duct cell. This is one of the pancreatic duct cells. We have here the lumen, we have the interstitial fluid, this is the basolateral membrane and the apical membrane. They are separated by the tight junction that surround the cells. As you can see here, again we have the muscarinic receptor for acetylcholine, and we have a different receptor. In acinar cells we have the CCK receptor. Here in the duct cell we have the receptor for the hormone secretin. And we have the sodium-potassium pump like any other cell in the body. And we have a different set of transporters. We have one antiporter for hydrogen and sodium. This is a secondary active transport system that is driven by the sodium gradient. And we have another transport system, which is sodium bicarbonate co-transporter. So the duct cell main function is to provide or release large amount of bicarbonate into the lumen of the pancreatic duct. So it should contain high concentration of bicarbonate. This comes either as carbon dioxide that will diffuse across the phospholipid bilayer, and under the action of carbonic anhydrase enzyme, carbonic acid will form. It will dissociate into bicarbonate that will be released into the lumen, and hydrogen that will be removed from the cell by this antiporter, the sodium hydrogen antiporter in the basolateral membrane. Another source for bicarbonate is this co-transporter, where sodium and bicarbonate are co-transported from the interstitial fluid into the plasma. Okay, now we have a cell that is not stimulated by secretin and acetylcholine. So what happens? This antiporter in the apical membrane, the chloride bicarbonate antiporter, will exchange chloride that comes from the fluid that is released by the acinar cells. We said acinar cells will produce a fluid containing enzymes and sodium chloride. So the chloride in the fluid coming from the acinar cells will be exchanged for bicarbonate. And this exchange is responsible for the low concentration of bicarbonate that is released into the pancreatic fluid at low flow rate in non-stimulated duct cells. But if the duct cell is stimulated by secretin and acetylcholine, this exchanger cannot cope with the amount of bicarbonate that is needed, because the amount of chloride that is available in the pancreatic fluid coming from the acinar cells is not enough for the release of large amount of bicarbonate. So stimulated cells will depend on another new transporter that will, under the effect of cyclic AMP, will cause the release of large amount of bicarbonate into the lumen of the duct cell or the pancreatic duct. I will explain all these steps in the coming 2 slides, inshallah. So the reciprocal change in chloride and bicarbonate concentrations, which increase in flow rate, may be accounted for the mixing of the 2 different secretions. We have sodium chloride solution produced by the acinar cells, and this predominates at low flow rates. And we have sodium bicarbonate solution produced by the duct cells. As I have previously explained in the previous slide, This predominates at high flow rate and dilutes the sodium chloride solution. Under stimulation, the amount of bicarbonate in the fluid released by the duct cell will dilute the sodium chloride. At high flow rates, the increased secretion of sodium bicarbonate leads to high concentration of bicarbonate and at low concentration of chloride in the pancreatic juice. As I said, with increasing flow rate, chloride concentration will decrease, bicarbonate concentration will increase in the pancreatic juice. The pancreatic duct cells can produce bicarbonate concentration of 140 to 150 millimolar. Bicarbonate inside duct cells is derived from bicarbonate transferred from the interstitial fluid by means of sodium bicarbonate cotransporter that is present in the basolateral membrane, as I have previously explained, or from the carbon dioxide that diffuses from the interstitial fluid, and then that carbon dioxide will react with water under the influence of carbonic anhydrase enzyme to form the carbonic acid. This one will dissociate into hydrogen and bicarbonate. Duct cells will export the hydrogen to the tissue or to the interstitial fluid in exchange for sodium moving down its concentration gradient into the duct cell. The hydrogen combines with bicarbonate in the interstitial fluid and will form carbon dioxide and water. So the cell can get rid of excess hydrogen that is produced from the dissociation of carbonic acid, and it will protect itself from the acidity produced by the hydrogen ions. So we have two mechanisms for the for the bicarbonate secretion from the duct cells. Now we have a duct cell that is rich in bicarbonate. Let's talk about how this bicarbonate will cross the apical membrane of the duct cell to arrive or to reach the lumen of the pancreatic duct. One mechanism is chloride dependent. So we have a chloride dependent mechanism, which means that the other mechanism will be chloride independent. The chloride dependent mechanism accounts for basal secretion of bicarbonate at low flow rate. There is an anion exchanger, which is chloride bicarbonate antiporter or exchanger that I already explained in the previous in the illustration. In the luminal membrane or apical membrane of the duct cell, this will transport bicarbonate out of the cell into the lumen in exchange for chloride uptake into the cell. Chloride is moving down its concentration gradient into the cell. So the driving force for this secondary transport system is the chloride gradient. And this chloride, as I said, is coming from the acinar cells that lie just proximal to the duct cells. The luminal chloride comes from the acinar cells fluid secretion and recycles through the channels in the apical membrane of the duct cell, as I explained. And the second mechanism is chloride independent mechanism. It accounts for bicarbonate secretion when the pancreatic flow or pancreatic juice flow rate is high. Stimulation of the duct cells with hormone like secretin leads to enhanced bicarbonate secretion. The supply of chloride from the acinar cells is insufficient to account for the concentration of bicarbonate achieved at high flow rate. So we should have another transporter, and this is the cystic fibrosis transmembrane conductance regulator, CFTR. This will serve as the pathway for bicarbonate secretion to the duct lumen when the duct cell is activated by secretin and acetylcholine. The lumen of duct cell, after the secretion of bicarbonate, will become electronegative with respect to the interstitial fluid. This will drag sodium. Sodium will enter the luminal fluid from the interstitial spaces by diffusion down its gradient through the paracellular pathway. Both sodium and bicarbonate will create an osmotic gradient. The resulting osmotic changes will draw water into the duct system to produce an isotonic secretion. So bicarbonate will move first. It will drag sodium after it. Both of them are osmotically active. They will drag water to produce an isotonic pancreatic secretion, at high flow rate. Now let's talk about the control of pancreatic secretion. We have pancreatic secretion does not become zero during the interdigestive period. The rate decreases and it fluctuates during the night. Then during the fed state, it will increase dramatically, and then after breakfast, and then will decrease back to noontime. Again, it will increase after lunch and so on. So the pancreatic secretion during the interdigestive period, the pancreas produces a basal secretion of enzymes and fluid. And as I said, the fluid is mainly water, sodium, and chloride. It increases in parallel with the intestinal migratory motility complex activity, and it is a function of the parasympathetic system aided by cholecystokinin. And both are responsible for coordinating this secretory activity. During the fed state, the controllers are different. The pancreatic secretion increases up to 20 times. 20 times increase in the volume of pancreatic secretion. It is regulated mainly by hormones, and these hormones are cholecystokinin that is triggered, or its release is triggered by the presence of fat and proteins in the chyme that is delivered to the duodenum. So in the duodenum, fat and proteins in the duodenum and the jejunum will stimulate the release of CCK from the eye cells or CCK cells in the intestinal glands. This will lead to secretion of enzyme-rich pancreatic juice. Secretin that is released in response to acid and to some extent carbohydrate in the duodenum. Both will stimulate the release of secretin from S cells in the intestinal glands. Secretin will stimulate secretion of aqueous bicarbonate-rich alkaline pancreatic juice. So CCK will mainly stimulate the acinar cells because they have the CCK receptor, while secretin will stimulate the duct cells that produce a large amount of pancreatic juice, which is very rich in bicarbonate. Again, weaker stimulus or stimulant for the pancreatic secretion during the fed state is the parasympathetic system through the vagus nerve and the acetylcholine released by the vagal efferents. So to summarize, as I said, fat and protein products in the duodenal lumen will stimulate the release of CCK from the duodenal mucosa. CCK carried via the blood to the pancreas or to the pancreatic acini will stimulate the pancreatic acinar cells to release enzymes or the pancreatic digestive enzymes into the duodenal lumen. And this will lead to the digestion of the stimulants. These are fat and proteins. On the other hand, the presence of acid and to some extent bicarbonate in the duodenal lumen will cause the release or will stimulate the release of secretin from the S cells in the duodenal mucosa or the duodenal glands. Secretin carried in the blood to the pancreatic duct cells will stimulate the release of large amount of aqueous bicarbonate-rich solution into the duodenal lumen, and this will dilute and neutralize the acidic chyme that was the stimulus for the secretion of secretin. Now let's talk about the phases of pancreatic secretion. As with the gastric juice, this occurs in 3 overlapping phases: the cephalic or cephalic phase, the gastric phase, and the intestinal phase. We have 3 phases for the pancreatic secretion. In the cephalic and gastric phases, information coming from the sight, taste, smell of food, thinking of food, or distension of the stomach, or chemical stimuli, so stimulation of mechano or chemoreceptors in the wall of the stomach. All of these will convey the signals to the dorsal motor nucleus of the vagus nerve, which in turn will convey signals or action potential to the parasympathetic ganglia and then to the wall of the stomach, where the release of gastrin-releasing peptide on the G cells will cause the release of gastrin. Gastrin will stimulate CCK1 or CCKA receptor on the acinar cells to stimulate enzyme release and small amount of water and sodium chloride. Also, the parasympathetic fibers will cause the release of acetylcholine at the in the basolateral membrane of the acinar cells, where we have the M3 subtype of receptors, muscarinic receptor subtype 3, and this again will stimulate the production or release of enzymes. Also, to some extent or to weaker extent, acetylcholine can stimulate M3 receptors on the basolateral membrane of the duct cells to stimulate the release of bicarbonate from the duct cells, or bicarbonate-rich fluid from the duct cells. And this pathway is for the cephalic and gastric phases. So the cephalic phase, the thought, sight, smell, taste, and chewing of food reflects vagal nerve stimulation of the pancreatic, acinar, and duct cells to secrete pancreatic juice through the pathway that I have already explained in the previous slide. The action of the vagus nerve is mediated by acetylcholine acting on muscarinic M3 receptors on the acinar and duct cells. Additionally, during the cephalic phase, vagal activity on the gastric antrum stimulates gastrin release, and gastrin has similar structures to cholecystokinin. So the gastrin release into the circulation acts on CCK1 or CCKA receptor on the acinar cells. So the vagal nerve activity produces a pancreatic juice that is rich in enzymes. As I said, the action is mainly on the acinar cells and to a lesser extent on the duct cells. Increased sympathetic splanchnic nerve activity will reduce the pancreatic juice secretion due to vasoconstriction mediated by norepinephrine, the mediator of the sympathetic activity. This norepinephrine acts via the alpha adenoreceptors on the vascular smooth muscles, reducing the blood flow to the pancreas, which will inhibit the secretion. Blood flow carries the amino acids, water, electrolytes that are components of the pancreatic juice. The gastric phase consists of neural and hormonal components that will stimulate the pancreas. The neural component via the gastropancreatic reflex. The distension of the stomach will stimulate stretch receptor in its wall. Afferent nerves and the vagus nerve will convey the information to the medulla, and this will lead to the excitation of vagal or efferent vagal nerve fibers to the pancreas. So the vagovagal reflex. This will lead to release of acetylcholine and stimulate the pancreatic secretion. Another part of the gastric phase is mediated by hormones. Protein digestion products in the stomach will stimulate gastrin release, and gastrin circulating in the blood will go to the basolateral membrane or will reach the basolateral membrane of the acinar cells that have the CCK1 receptors. So they will be stimulated to release the digestive enzymes. Regarding the intestinal phase, here the intestinal phase, the source of the stimulus is present in the lumen of the duodenum. That's why it is called the intestinal phase. Fat, protein, acidity, they will stimulate the release of cholecystokinin for fat and protein, and secretin in case of acid, and to less extent bicarbonate. The released hormones, either secretin or cholecystokinin, will lead to the stimulation of acinar cells and duct cells to produce enzymes as well as fluid that is rich in bicarbonate. So the intestinal content or intestinal contents play a major role in controlling pancreatic secretion. The intestinal phase consists of hormonal, mainly cholecystokinin and secretin, and neural, which is called the enteropancreatic vagovagal reflex components, including stimulating the pancreas. The hormonal are the presence of monoglycerides, free fatty acids, amino acids, and peptides in the duodenum, but not bicarbonate nor acid. In the duodenum will lead to the release of cholecystokinin from the eye cells in the duodenal mucosa. The released cholecystokinin will enter the circulation and will be delivered to the pancreas. There it will stimulate enzyme secretion via CCK1 receptor on the basolateral membrane of acinar cells. The presence of HCl in the chyme that is delivered to the small intestine from the stomach during gastric emptying will lead to the stimulation of secretin released from the S cells. Secretin will enter the circulation. Again, it will be delivered to the pancreatic duct cells and it will stimulate bicarbonate and fluid secretion. Please notice that cholecystokinin alone has weak effects on fluid secretion, but it potentiates the action of secretin. So CCK alone has very weak effect on the duct cells, but if secretin is present, then cholecystokinin will potentiate the effect of secretin. The neural component of the intestinal phase is caused by the presence of lipid, peptides, and amino acids in the small intestine. These will stimulate the enteropancreatic reflex. Again, it is a vagovagal reflex mediated by vagal afferents and efferents via the medulla, with afferent and efferent pathways in the vagal nerve. This will stimulate the pancreatic secretion. Now, what are the interactions between acetylcholine, cholecystokinin, and secretin? The three major or main players in pancreatic secretion in the different phases of pancreatic secretion. The interactions between acetylcholine, cholecystokinin, and secretin at the cellular level are important in determining the response of the pancreas to a meal. So it affects the quality of the pancreatic juice, whether enzyme-rich or bicarbonate-rich. The effect of cholecystokinin or secretin in the presence of acetylcholine on pancreatic secretion is much more potent than the effect of cholecystokinin or secretin in the absence of acetylcholine. So there is potentiation or synergism. The potentiation phenomenon is due to the use of different second messengers by these different mediators. IP3 and diacylglycerol or DAG are the second messengers mediating acetylcholine and CCK effects, while cyclic AMP is the second messenger mediating secretin effect. When hormones or neurotransmitters use different mechanism or different mediators or different messengers, second messengers, then the combined effect of them will be synergism and or potentiation. What inhibits pancreatic secretion? Once maximally stimulated, pancreatic secretion begins to decrease after several hours. Regulatory mechanisms responsible for feedback inhibition of the pancreatic secretion to its basal interdigestive state include the presence of fat in the distal end of the small intestine, in the ileum and colon. This will stimulate the release of peptide YY or PYY from the L cells present in the glands of the terminal ileum and the colon. This hormone will reduce pancreatic secretion by acting on inhibitory neural pathways, as well as by decreasing pancreatic blood flow. Somatostatin from these cells and glucagon from the pancreatic alpha cells may also be factors in returning the pancreatic secretion to the interdigestive state after a meal. The references that were used for today's lecture are these 5 references. Thank you very much for your patience, and I hope that you got some benefit from the lecture, and I welcome any questions or any clarifications that you need by email. Thank you, stay safe, and good luck.